Last ed puzzle, what? Last ed puzzle, what? All right, cool. That happened. All right, let's get into chapter 17, which is going to be talking about human health and environmental risk factors. So there's 3 categories of human health risk, essentially 3 different types of groupings that we can fall, anything that can harm humankind. Biological, which would be when we think about diseases or infections. Physical, so natural disasters, tornadoes, hurricanes, flood, volcano, and chemical. So chemical could be anything that is chemical in nature. So when we're talking about like endocrine disruptors, if we're talking about toxic waste, this is all going to be chemical. We're really going to be focusing on just these 2. I'm going to talk about biological first and then chemical as well. And then we're going to talk a little bit about some super fun legislation, and then we're going to be done. All right, so as promised, biological risk first. Infectious diseases are going to be caused by infectious agents. Hakuna Matata. I don't know why I said that, but that makes sense. These are also known as pathogens. These are going to be the biggest cause of human deaths. We're going to take a look at that in just a second. So infectious diseases, if I'm looking at everything on over here on the left that's affecting humans or causing human mortality, infectious disease is about 26%. Although cardiovascular disease is greater. We'll come back to that. All right. So when we get into that, we can break this down into different subcategories of all these different types of infectious disease. We can look at respiratory ones, HIV. These are those that are sexually transmitted. We can talk about stomach or intestinal infections, ones that are coming more from like a virus. And we can also look at those that are being caused by bacteria or our good friends, worms. If you forget, like ringworm, yay. Really good stuff. All righty. There's also 2 subcategories. So biological risks was a subcategory of human health hazards. It was biological, chemical, and nature, physical. Biological risk can then be brought down into chronic or acute. Chronic, think about like, I have like a chronic headache. If someone says they have a chronic headache, that means that they've had this headache for a really long time, right? So chronic disease is something that slowly impairs the body's function over a very long period of time. So something like HIV/AIDS, like you can get HIV and then that virus can be in, the lysogenic long-term life cycle, and that HIV virus can be dormant for like decades. And then like 40 years later, like turn into AIDS. So that is something that is a long-term, many decades long. We would consider HIV and AIDS to be a chronic disease. Acute diseases, there's going to be short-term ones. So these are ones that rapidly occur. So usually in the span of a couple days, a couple weeks. So you go swimming somewhere and there is a really weird bacteria in the water, and then you drink the water, and then that bacteria has a little fiesta in your body and causes you stomach cramps and intestinal bleeding and diarrhea. Good morning or good afternoon, whatever time you're watching this video. So that would be something short term, and it happens very quickly in the span of like a couple days, a couple hours, a couple weeks. All righty. When we're looking at the leading risks of health problems worldwide, especially those risks, we can really see a huge difference in what is actually causing the greatest amount of deaths and injuries for individuals, whether they're in a low-income country or in a high-income country. Now, there are a couple similarities, like high blood pressure is number 2 for both. Tobacco use, I guess, is actually pretty different. So we've got a lot of similar root causes or root risks that are in common, but the difference that we're seeing is what are going to be our prominent ones. So for high-income countries, you would think of maybe like tobacco use is kind of a leisurely thing. I mean, especially if you're thinking back like in like the 60s and the 50s, and even before that, tobacco use was huge. I mean, it was like all publicized. And so that was one of the highest risk factors, and that could lead to death in our high income countries because we had the funds, we had the leisurely time, maybe. Not always, but could happen. Whereas when we're looking at like low rates during childhood, anemia, malnutrition, all those could be different things that could occur in a low income country. So we're starting to see these differences depending on where an individual is born and what is going to be their most prominent risk factor that they're facing throughout their lifetime. High blood glucose, again, these are some that are going to be pretty similar, but we're really seeing that difference in that tobacco use and that low weight during childhood being those leading factors. Historically, we had the plague. These are all going to be biological risk factors, the plague, malaria, and tuberculosis. It's interesting. I always think whenever we're looking at this, because we can see that all three of these were organic in nature. So none of these were virally caused, whereas I feel like if there was one more and there was one for our generation, it would probably be like COVID-19 as like a historical pandemic for us. But back in the day, it was all bacteria, protists, and other living things. So these are all organically based historical diseases. I would be familiar with them. Your textbook does a really good job talking about the history of these, how they've actually spread between one individual to another. I think one of the key things is that with all three of these, they're highly infectious, highly contagious. Again, things that you've probably heard of and seen of is if we have a high infection rate and a high contagious factor for a different disease, that's going to cause it to spread wide scale in a very short amount of time, leading to something that we would consider potentially a pandemic. Sometimes you might hear epidemic versus pandemic thrown around. I wanted to just quickly talk about that. Epidemic is usually when something's on a smaller scale than a pandemic. So a pandemic is going to be something like COVID-19, infected many, many countries, widespread, highly contagious. So because it was such a broad scale and affected many different regions, we would consider that a pandemic. Whenever I think of an epidemic, we're usually thinking about something that's either isolated to a small area or to a country in itself. Something that is transmissible, we're able to isolate it, and it's not as widespread globally as like something like a pandemic. All right. Emergent diseases. Again, your textbook was written before the good old COVID-19, so it is not on here, but I guarantee it would be on here if it was. HIV and AIDS, interestingly enough, they've actually found HIV and AIDS in a species, I want to say it was chimpanzee, definitely one of our primates, which leads us to believe that potentially HIV and AIDS might have originated in some of our primate relatives, and then it might have been transmitted to humans, and that might be how it actually emerged and infected and became part of our human life. Ebola, I'm sure you guys have heard of Ebola as well, transmitted virus. A lot of these actually, interestingly, the last time was all organic. These are all viruses in nature. Avian flu or bird flu is very similar to the human flu that affects us. So you've probably heard, especially hearing about like COVID-19, that is an H1N1 is our influenza. Influenza is H1N1. This is a different variant, but avian flu H5N1, but it still has a lot of similar characteristics. That's why they're both considered flus. They're in that influenza family, but there are some differences. However, because there's a lot of similarities, that this H5N1 was transmissible to humans because we have a lot of similar receptors for it. Yay, humanity. Mad cow disease. Bad stuff. So just to kind of like summarize a lot of this is talking about the pathways of transmitting pathogens. Pathogen is anything that is going to be harmfully causing or harmfully causes harmful effects to humans. So we can get things from water, we can get things that's airborne, we get drinking water in the air, we can get things in our food if something's not prepared, or if it was prepared a long time ago and it has now gone bad and we got some bacteria in there. Wild animals can transmit to us, domestic animals as well. What's interesting though is that the only one where we are giving it to somebody else is other humans. So if I got something, I can give it to Billy Bob Joe. However, I probably talked about it a little bit with you guys. Like, I'm not going to go pet my pet turtle Shelly, and like she's not going to get COVID-19 from me. So a lot of these viruses are going to be ones that are selectively affecting other humans, but we cannot transmit to wild animals. Sometimes it's even like in the case of like in the Black Plague, where wild animals were carriers, they were not themselves affected by it. So rats were a huge carrier. It was actually on their fur, that plague virus. No, plague bacteria, sorry about that. So they were carriers of it. They transmitted it to humans, but we were not giving it back to the rats, if that makes sense, because it was something that was specific to humankind. All right, so that was all biological, living things, infections, and diseases. Chemical risk is going to be the other one that we're focusing about in our class, environmental science. Chemical risk sources can be in our medication, pharmaceuticals, that if maybe we change up that molecular formula, something doesn't go quite right, we accidentally increase the dosage too much, that can be a chemical risk. Insecticides, lots and lots of toxic chemicals. We could have ones that are like disruptive to protein synthesis, disruptive to DNA sequencing. In our insecticides, herbicides, and fungicides, which is why, kind of thinking back to land use in agriculture, the more that we can do of integrated pest management, the more that we can do of organic or natural killers, the better. Of course, these are super, super effective and a lot cheaper. So, I mean, win some, you lose some. Cleaning products as well. Again, anything that is chemical in nature could become potentially toxic or lethal. If you accidentally, like, I don't know, you're wiping down your windows with the window cleaner and then you lick your hand. If that's something you do in your free time, you could potentially be poisoned from whatever chemicals are in those cleaning products. And of course, industrial chemicals as well. All right, there's a lot of stuff going on here. So when we're actually looking at what could then occur from them, so this last slide was the sources. What results from them is going to be the risk that could be associated with those different products. So some things that you could get from those chemical sources would be things like neurotoxins, anything that disrupts your central or your peripheral nervous system. So if it causes paralysis, if it causes detriments to your brain functioning, if it causes any detriments to maybe, I don't know, your spinal cord or your sensory organs, that's going to be a nervous system toxin. Carcinogens, that carcino is a root word for cancer. I don't know, they've got a couple letters in common. Carcinogens could cause us cancer. I never can say this. Another one of those teratogens. These are gonna be ones that can affect the development of embryos or fetuses. Obviously, those are our most vulnerable individuals of humanity. So we want to protect them as much as possible and try to avoid exposure to those. Allergens, these can be anything from a minor allergic reaction, just a couple sneezes, to a massive or major allergic reaction where an individual's throat could swell up, they could have hives everywhere. So we could have a huge range of allergic reactions. And endocrine disruptors. Now we're going to take a day, some point this week, before the STAR test, to talk about endocrine disruptors and how detrimental these can be for us. So hormones are incredibly important for us. Not only, like, obviously, I feel like the go-to ones we think about are estrogen and testosterone, are sex determining and sex influencing hormones. But even like my all-time favorite hormone, if it's not weird to have a hormone, is called ghrelin. Ghrelin makes you growl, as the way I always remember it. So ghrelin is a hormone that whenever an individual is hungry, your pituitary gland is going to release ghrelin. That tells your stomach to go, wah, 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 and it sends you a signal like, hey, put some food in me. So ghrelin's a great one. Growth hormone helps you grow. But a lot of these, if we disrupt, if we either overstimulate or understimulate our hormonal receptors, we could have very drastic results towards those individuals, since our endocrine system is our hormones, and those are our chemical messengers for the entire body. I found this little graphic, I thought it would be helpful. So there's two main ways that endocrine disruptors can happen. All right. So normally we've got a hormone, it's going to go bind to the receptor and cause a response. I am hungry, I'm going to release ghrelin. Ghrelin activates my stomach receptors. My stomach is going to growl, telling me, hey, give me some food in here. We could have hormone mimics, that's a type of disruptor, and hormone blockers. These are our two endocrine disruptors. Hormone mimic is going to overstimulate. So even when I'm not hungry, this is maybe food's not a good example, because I know there's a lot of there's a lot of disorders that also tie in with food, like overeating and undereating with that as well. But whatever hormone it may be, whether it's estrogen or testosterone, if we have a hormone mimic, that can overstimulate something. So there is some studies that are actually, if sea turtles, I don't remember if it's sea turtles, there's some aquatic animals where they're actually seeing a huge increase in female individuals in that population because of these hormone disruptors. These individuals are seeing influxes of estrogen, and it's causing sex changes in a lot of these organisms. So this would be an example of an endocrine disruptor. I'm having an overstimulation of a particular endocrine, and that's having an effect on the organism as a whole. Endocrine disruptors can also be hormone blockers. Hormone blockers actually are going to essentially put a plug on the receptor. So instead of actually stimulating the receptor, they're going to bind to the receptor and block the actual, not antigen, the actual proteins that should be binding to that receptor are going to be blocked and will not be able to actually adhere to that receptor. So this one, we have a decrease or a ceasing of that response. Again, very detrimental to an organism who relies, many of their body processes rely on these hormones being in balance. All right, you might remember LD50 from biomagnification. We're coming back to it because it's really important when we get into these chemical concentrations and what is actually okay to put into our environment and where is the limit of having these in our environments. So LD50 stands for lethal dose 50. If you may recall, or if not, we're learning it again right now. LD50 is going to be the dosage, usually in milligrams per kilogram, that is required or would result in mortality of 50% of the population. So if, let's say I've got a population of 100 rats, I give them I give all of them 75 milligrams of insert bad chemical here, and then 50% of my rats die, but 50% live. That means that the LD50 for that population is what amount I gave them, 75 milligrams. The ED50 is the effective dose that causes 50% of the animals to display harmful but non-lethal effects. So while LD50 is what amount we need in order to essentially have the population is going to be killed off, ED50 is going to say this is the dosage where 50% of the population is showing bad side effects, though they're not dying. So usually the ED50 is going to be a lower amount than the LD, because the LD is going to be a little bit more because it's going to, unfortunately take the next step and cause the death of the individual. ED is going to cause bad side effects, but not result in that fatality for 50%. So ED50 is usually a little bit less, a little bit more conservative. All right, so when we're actually looking at these LD and ED50 studies, we're going to be looking for something known as the threshold. That threshold is like what I talked about with the rats, that's 75 milligrams. This is the dose at which the effects can actually be detected. Thank goodness we don't do these labs or studies on humans. Woo! Not great that we're still using living things, but usually mice and rats, because they have a lot of similar anatomy to humans. They're fellow mammals to us, so we've got a lot of similar processes going on. These are usually what we're going to be using for these studies. And I will say, just kind of from like a research standpoint, because y'all know I used to do research for a while, they do the 3 R's as well: reduce, reuse, and refine. So scientists are using the least amount possible, and they're using the least complex organisms possible for these studies. I know it's hard to think about like killing animals for science, but they are, I mean, the committees are very extensive to make sure that we're making this as ethical as we can. So usually we'll see an S-shaped curve that I'm going to show you guys on the next slide. And this is going to tell us the relative toxicity of chemicals. So here is my LD curve. So I'm seeing dose. Usually it's in milligrams per kilogram or grams per kilogram of organism. But this is going to show my percent mortality. So we're going to see an S-shaped curve. We're going to start out pretty low. Low dosages usually don't kill a whole bunch of people. As we start getting upwards, we might be getting closer to the ED50. That ED50 being, we're seeing some harmful effects in 50% of the population, but no one's really dying yet. I guess we have like 10% of the population dying. And then we get up to the LD50, the lethal dose. 50% of my population is facing mortality. This is going to be my threshold. That is my LD50. And then finally up at the top, I mean, too much of anything can be a bad thing. So eventually too much of something is going to cause nearly 100% mortality. All right, so that's lethal dose. Oh my gosh, that honestly scared me. I literally jumped. Oh my gosh, sorry. Oh my gosh. All right, anyways, that was a horrifying face swap. But okay, as you would hope, we are monitoring chemicals, and we're monitoring our water, and we're monitoring all these different products that we are putting into our households to make sure that we're never exceeding that lethal dose as much as possible. The EPA, the Environmental Protection Agency, is going to be the ones who are in charge of regulating this. The Toxic Substances Control Act of 1976 and the Federal Insecticide, Fungicide, and Rodenticide Act are both going to be looking at these different chemicals and making sure that we're monitoring them, identifying that LD50 for them, and making sure that whenever we're producing any product that has these chemicals, that we're ensuring that they are well below that LD50 threshold. Obviously, we care about humans, so we want to make sure that as few as possible are being negatively affected. So it is relieving to hear that we have these two different acts that are going to be in charge of protecting individuals and ensuring their safety. Kind of like in energy, we talked about cogeneration of energy. When I have one process, let's say that's producing electricity, heat is produced as a byproduct, and then that heat byproduct was then used to fuel more energy. So I had two sources of energy coming from one process. That was I already forgot what it's called. Cogeneration. Oh my gosh. Okay, sorry. Cogeneration. It's like 5 o'clock. It's time for me to go home. Synergistic is be very similar, but in terms of risk factors. So if I have two things that are happening at the same time, when one thing causes you harm and two things causes you harm, but having them both at the exact same time causes you even greater harm than one or the two would. So sometimes whenever I think about this, I think about comorbidities. When an individual has two chronic disorders, and sometimes those two chronic disorders just happen, like it's an unfortunate lot of luck where those two comorbidities actually interact in a way where it's even more detrimental. It's almost like they negatively feed off of each other. So we got an example here. If I have a carcinogen, a cancer causing substance such as asbestos, this is going to be much higher if, like, the health impact is going to be much higher if an individual also smokes tobacco. So smoking tobacco can cause you lots and lots of respiratory impairment. Asbestos, also respiratory impairment, but also it's a carcinogen. Then these two effects are then multiplied because an individual is exposed to both asbestos and they're also smoking tobacco. So again, we can call those comorbidities can fall into this subcategory here. Sorry, I feel like I'm just like, go. Okay, how can we be exposed? This is very similar to when we talked about rates or pathways of transmission of our biological disease. Again, this is now we're talking about chemicals. This is talking about this chemical risk. We can get them from our food if they're prepared differently, or maybe when in the sterilization process, or maybe they were preserved really weird and there's some weird, strange chemical that's on my food. I can get chemicals that are airborne from factories, such as toxic ash. We can get chemicals from our water. If it's, let's say, fertilizer, eutrophication, I guess not eutrophication, but fertilizer runoff, I should say, that fertilizers, chemicals can get into our water. If there's a faulty process in the wastewater treatment plant, and whenever I get that fresh water, for some reason, some of those chemicals in the fertilizer got into my water supply, I can obtain it that way. Hopefully you're not eating soil, but definitely wash your fruits and vegetables when you get them, because you could get chemicals from the fertilizers in your soil. So wash all of your produce when you get it. And this is also showing that, unfortunately, in women, some of these side effects can then also be passed on to the fetus of an individual, especially if they are pregnant at the time of exposure. So always sad things in this class. I'm so sorry. Speaking of sad things, here's more sadness. We talk about bioaccumulation. This should hopefully be a review from when we talked about land use, agriculture, I don't remember, water. At the time we learned about this. Bioaccumulation is the increased concentration of a chemical within one organism. So this is something that's not going to be digesting out. So for example, DDT is a big one. So DDT is not something that whenever I ingest it, I'm going to digest and then poop it out. DDT is something that's going to actually get into my tissues, and it's going to bioaccumulate in my tissues. So bioaccumulation is the aggregation or increased concentration of one chemical in one organism over time. Bioaccumulation can then lead to biomagnification. So if I have bioaccumulation of DDT in the plankton, it's not leaving the plankton. The DDT in one plankton is going to biomagnify up the food chain, because each of these individuals are eating more than one prey, if that makes sense. So my 3-spined stickleback might be eating, like, I don't know, it looks like if I'm doing the math on here, they're eating 5 phytoplankton or 5 plankton. So they end up getting even more DDT. This flounder looks like it's eating about 5 of the 3 spine stickleback. So it's getting more DDT. And the osprey is eating about 10 of the flounders. So the osprey, again, because its DDT is accumulating and staying in the tissues, when one organism eats multiple of its prey and they all contain that DDT, this is going to biomagnify as we move up the food chain. Hopefully that's all review. Remember the paper clips. Remember that frustrating paper clip lab that you all love so much. Okay, we're getting close to the end, I promise. Persistence is gonna be how long a chemical remains in the environment. This is gonna be tied right in with the half-life of a chemical. Half-life you might have learned in chemistry. If not, we're learning it now. Half-life is going to be the amount of time that a particular substance takes to decay to half of its original size, weight, or volume. So for example, radon, if it's 4 days in the air, let's say I have 100 grams of radon floating through the air. In 4 days, I'm now only going to have 50 grams of radon. And then in 4 more days, I'm going to have 25 grams of radon. And then I forgot where I was at. So hopefully you got that. This is going to be for a given amount of half-life, whatever volume I have of a particular substance, it's going to shrink down or decay and break down until it is only 50% of its original size, mass, or volume. This is going to be extremely important to know, especially in regards to the LD and whenever things are running off into our soil. I want to know how long is something going to stay so that I need to worry about it. So DDT, 30 years for it just to come down 50%. That doesn't mean that DDT goes away completely in 30 years. That means I go from, let's say, 100 grams to 50 grams. And then 30 more years go by. And now I'm down to 25 and then 12.5. But at this point, now we're at 100 years or so. And now I'm only at like 20, let's say 120 years would get me to 12.5 grams. So, and that's like, I don't know, 1/8 of the original size. And it takes 120 years. So what we're seeing for this is that we need to know the half-life of different chemicals and different environmental risks so that I can try to figure out, okay, this one takes a really long time to break down. What can I do to mitigate how much it actually gets into my soil, how much it gets into my water, because I know this is something that's going to be persistent. This insecticide, because its half-life is only a day, I probably should be worried about it, but not as worried as I would be about DDT, because this one is not going to be very persistent, and it's probably going to kind of environmentally self-decompose and self-decay, and I don't really need to spend all my time and energy on this one compared to PCPs and DDT. Hopefully that makes a little bit of sense. I'd be happy to explain more if not. I think we're going to talk about it in class anyways. All right, last, I think 3 things. When we're actually trying to figure out risk and we're looking at risks holistically, I'm going to be looking at risk in 3 different ways. I'm going to first assess the risk. Then I'm going to try to figure out what is my acceptable level of the risk. Then I'm going to try to manage that risk. So first I want to do assessment. So I want to figure out, what is this hazard? What is its LD? What is the extent of exposure that is necessary in order for it to be transmissible? When we're looking at this risk assessment, we're also going to be looking at it quantitatively and qualitatively. So when I'm identifying a hazard and we're actually trying to figure out what actual harm could this do to us, I want to look at it in terms of quantitative risk, which is the probability that I'm actually going to be exposed to this hazard, multiplied by the probability of actually being harmed. So this can be as simple as even being in an automobile accident, right? So the probability, I'm sure there's a statistic out there that I don't know. I'm sure there's a stat that says this is your probability of being in a car crash in your lifetime. Okay. Then we can multiply that times the probability of me being injured, or hopefully not, I hope this never happens to any of us, but killed from that car accident. I would multiply the risk of me being in a car crash times being actually harmed by that car crash, or it might just be cosmetic damage to my car. We wouldn't really include that. That's going to give me my overall assessment of how bad is this risk. All right. We can also evaluate qualitatively, and I'm sorry that bottom left corner is super, super hard to see. This is when we're kind of like objectively looking at it, not objectively, subjectively looking at a risk and saying, is this going to cause me harm or not? So if I'm looking at it and I say, okay, it is raining outside, I can use my mind and make a hypothesis that it's going to be more likely that I could get in an accident due to skidding or hydroplaning. This is something I know. There's no math involved, there's no statistics involved, but I just know from my lifetime experiences, this is a risk that could happen in a given situation. Once I've assessed the risk, then I'm going to move on to risk acceptance. Oh, sorry, I forgot. This is some of that risk assessment. So actually getting that data. So this is all the likelihood that somebody, it's potentially blocked, it's blocked on my screen, that an individual could unfortunately be killed from this in their lifetime. So this would be a quantitative because it has to do with those numbers. And this is all calculated or used to help calculate my risk assessment, my probability of being exposed multiplied by the probability of actually being harmed. All right. Once I figured out this is my risk, this is the probability that is involved with it actually affecting me, then I want to figure out, okay, what's that LD? What's that lethal dose? What is the level of risk that I can actually be tolerated? So this is when we get on our LD 50, when we start looking at our ED 50, effective dose 50, ED 50. Once I've said, okay, this is my toxic substance, this is the level of acceptable range that I'm actually willing to expose people to, then I say, okay, if I know my harmful substance, if I know how much or how little I want it to actually be exposed to people, now I want to actually manage that risk. I want to see what can I do to try to prevent this from affecting individuals. And I really want to take into consideration, what are the economics at play in this? What are the social, ethical, and political issues? So if there is something that is coming from like railroad tracks, and it is more likely that individuals who are of lower income happen to be next to railroad tracks, and there's a whole bunch of politics involved with that, and a whole bunch of really fun history, not really, but involved with that, I'm going to know, okay, my low income families are more at risk for this. So I want to make sure there's things in place to protect those individuals. So again, we want to assess and know the risk. We want to accept the level and figure out, what do we need to keep this risk below a certain threshold? Then I want to put in some policies, and I want to put in some plans to help protect individuals from these risks. I feel like it's a marathon of talking. Okay, this is literally like, oh, heck yeah. Yeah, we got like 2 more slides. Okay, so when we're actually looking at products, there's another way that we can actually look at different ways of managing risk. And this is in products, but it also can be found, mostly it's actually just like products, yeah. So we can do the innocent until proven guilty principle of managing risk and preventing risk occurring to animals. Oh my gosh, people, I guess we're also animals. Or we can do the precautionary principle. Each of these are both attempting to mitigate risk and to protect human beings. This one is probably the fastest, the innocent until proven guilty, because essentially what we're doing is we're reducing process. We do some research. We want to make sure things are good. We do some testing, but we're kind of just like, it's good. Get that product out there. Money, 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 money. We get that product out there, but then we're like, oh crap. We're realizing, product orange over here is actually unsafe for humans. Ugh, take that off the market. We get it off of the market because it's not good. But this one is actually okay. So we're like, all right, keep the safe one. So this is the everything's fine until we find out that there's a problem later. Obviously, entails people actually getting harmed from it. The more cautious, conservative, safer option is going to be the precautionary principle. In this one, once we develop a product, we do extensive pre-marketing testing, and we realize, okay, man, orange products, before it's even hit the shelf, this is not a good product. Look at all this. There's like carcinogens in here. Oh my gosh, there's endocrine disruptors. Get this off the shelf. Make sure it does not get sold. So only the safest ones actually make it to market. The ones that are unsafe do not even make it out of testing. So again, this is probably a lot more money intensive. It's a lot more time intensive. You have a much more delayed feedback in terms of monetary gain, because you're not putting it out on the market as fast, but you end up with probably less lawsuits, and you probably have less customer complaints if you find out that your product is actually unsafe. Oh, yes, we're on the last slide. We can do it. Stockholm Convention, 2001. Not in your lifetime, but that's okay. It's in my lifetime. Woo. Stockholm Convention, a group of 127 nations, beautiful, gather in Stockholm, Sweden. And essentially they identified 12 chemicals called the Dirty Dozen. And they realized, okay, PCBs, not great. DDTs, not great either. These especially have a really long half-lives as well. So when they realized, okay, these chemicals are causing horrendous side effects. These are causing lots of infections, diseases, and chronic illnesses in individuals. What they did is they tried to ban out, phase out, and reduce these 12 chemicals. And a lot of these especially are manufacturing byproducts that probably arose during the Industrial Revolution. DDT, I always think about whenever we're thinking about like insecticides, herbicides, it works really well, but it also affects humans as well. A lot of these were also endocrine disruptors. What's beautiful about this is that 127 countries all came together and said, this is not good. We need to do something for the sake of humanity. And they like got united together and they decided, yeah, we all agreed to ban out, phase out, and reduce each of these products, and really improved a lot of quality of life, at least in this particular vein of human life. So that at least, I'm happy for once we got to end on a happy note, especially because we're talking about like death, disease, and sadness and illness. Riddle me this. A man is looking at a photograph of someone. His friend asks who it is. The man replies, this person, brothers and sisters, I have none, but that man's father is my father's son. Who is in the photograph? Think about it. I'll let you know in class, probably, unless I forget. I hope you have a fantastic day. Congratulations for making it to your very last Ed puzzle. I hope you have a fabulous day, and I look forward to seeing you guys in class. Thank you guys. Bye-bye.